forgotten, I'm still Dr. Wyman. That is not Dr. Wyman.
Not bald or short.
Jennifer Robeson came on up this morning to give a talk on her effort to make soybeans tougher. You ever dealt with soybeans? They're pretty tough now. You want to make them tougher. Like you. All right. Jennifer's had a unique journey. She started out at Dickinson College. That was in Pennsylvania, right? All right, where she was a bio major, chem minor, and she worked with invasive species. All that weeds. And then she decided she didn't want to do with weeds anymore, so she moved to the coast, University of Delaware. She worked on marine life. Ooh, fish.
Not fish, coral.
Was it fish?
It was coral.
Coral.
Yeah.
Better than fish.
Better than fish. You have to go to the tropics.
And she worked on her master's degree there, and then she took a hiatus from science for a bit, and she did some other things. You can ask her about that if you want. And then she came back to her senses and realized, plants are much better. Much better. No salt water, sunscreen as much, not so bad. And so she's been working on soybeans at Pui, Indiana University of Indianapolis. So she's a jaguar, so kind of our cousin. Cougars, jaguars, big teeth, bite you. I met Jennifer
3.
years ago.
Yeah.
At an American Society of Plant Biologists conference that she and I attended. And one of the best parts about conferences is you get to meet people you don't know. It's called making friends. You do that in your core classes. We do that as scientists at meetings. And so we immediately met, we hit it off immediately. You'll find we have a lot of similarities in personality. She's an absolute stellar human being. I encourage you to talk to her. She's done stuff. She knows stuff, and she wants to talk to you. She thinks it'd be a great talk. So I'm done. Jennifer, you're on.
Go. Thank you, Aaron, for that kind introduction. Today, I'm going to tell you about our quest to try and make soybean more cold tolerant. So right now, when it gets a little cold outside, soybeans freak out, and if they're little baby soybeans, they die. If they're older soybeans, they don't make babies, so we don't get food, right? Because we eat soybean seeds. So it's very, very important to understand soybean. Soybean are one of many crop species around the world, and whether we're directly eating the plants or we're using the plants to feed the meat that we're going to be eating, they're the backbone of food production. This map is showing all the croplands of the earth, and the darker it is, the more area that's used for crops. This is the Midwest, right? See the mitten, that's Michigan. Very, very dark. We do a lot of crops around here. Other areas of the world, like pretty much all of Australia, is very light, not a lot of crop productions. So because of global food production and our global economy, this food has to be moved around. And right now, worldwide food production, which is in the red, is tracking pretty well with consumption. So you've got blue lines, and this is just 3 major crop species: corn, wheat, and rice. At the moment, we're tending to meet demand. However, there are some factors at play that in the near future, we might not be able to meet the demands of our world. And one of those is environmental stress. Animals, us, it gets cold, we put on a sweater, we run away, we go sit by the fire. It gets hot, we put on a tank top, we get out the fan. Plants can't, right? Plants are rooted in place, and they have to confront these changes as they occur. In a single day, you can have bright sunshine and pouring down rain. In a season, you go from 40 degrees to 90 degrees.
Sounds like Michigan.
In a day, right? Like you guys do this every day and you run away. Think about your trees that have to sit there and just take it. So these are all pictures of soybean under some pretty extreme drought, flooding, heat stress, they're all wilted, and then cold stress, there's a bunch of tissue damage. They have to know how to adapt to both long and short-term exposures. We're trying to understand how do they do this, right? For scientists, that's really important to know how do they adapt and how can we help them adapt. Why we want to know about that is by 2050, they're estimating there'll be a 25 to 50% decrease in agricultural yield, which means by 2050, we might be able to make 50% less food than we're currently making. And if you think back to that production and consumption, it's going hand in hand right now. If we were to lose 50% of that production, I don't think we're going to be real happy eating 50% less food. So what are we going to do about that? Anywhere that's red is losing a lot. And there again is our little mitten. There's Michigan. Michigan's red. It's going to be losing a lot of agricultural yield in the next couple decades. So we're trying to figure out what can we do about this. On one hand, we have agriculture loss. On the other hand, we have world population. World population is continuing to increase. By 2050, we're going to have 9 billion people on this planet. And we're going to be producing 50% less food. If we took all the food that we produced in 2009 per person, made it a per person value, and then spread that same amount of food to the amount of people we will have in 2050, you get an 800 calorie per person drop in the amount of food. Right? So this is a significant amount of food that's going to be lost. So understanding how plants and crops in particular change to these environmental impacts is super, super important. My environmental impact of choice is cold. So I'm looking at freezing and cold tolerance of soybean. When we think about cold in plants, we separate them into two very broad categories. You have cold tolerant plants, and these plants are able to undergo molecular and physiological changes to survive cold and subsequent freezing temperatures. And the little plant labrat, Arabidopsis, it is cold tolerant. So we know a lot about the cold tolerant pathway because we know a lot about Arabidopsis. Other plants are cold intolerant. They're unable to undergo molecular and physiological changes. They can't adapt to cold. They can't adapt to subsequent freezing. And my friend the soybean, who's sad over here because it's snowing on him, is one of those cold intolerant plants. When we think about cold, there's a process called cold acclimation. Arabidopsis is very, very good at it. Cold acclimation is when you're exposed to a low cold temperature and then subsequently exposed to really, really cold temperatures, you survive better. Think about this from a time standpoint. It gets bursts of cold in the fall, right? And then we have that hard freeze. Those bursts of colds are signal for the plants to say, hey, something worse might be coming, get ready. And Arabidopsis is really good at that. In fact, if we look at this plate, here's some Arabidopsis that were held for 2 days at 4 degrees C, and then this plate was kept 2 days at 20 degrees C, and then we shoved them at negative 10 degrees C for a couple hours, and then pulled them out and watched if they would recover. Obviously, the ones that had the refrigerator temperature, they're green, look pretty healthy. The ones that were kept at room temperature, not so much. So this is cold acclimation. You give them a little burst of cold, they survive cold later, right? Even worse, this is negative 10. And they survive negative 10 a whole lot better than their non-acclimated counterparts. This is Arabidopsis. My story is about soybean. All right, so we're using Arabidopsis to compare what's happening in soybean. And in Arabidopsis, we know that the underlying molecular mechanism is called the CBF cold responsive pathway. And this is just a quick representation of the pathway. Basically, when plants sense cold, membranes get more rigid, right? Less flexibility in the membranes. Cytosolic calcium levels rise. This turns on a whole bunch of kinase cascades. In this case, it leads to the activation of a protein called Size1. It stabilizes ICE1, so it keeps ICE1 from being broken down. ICE1's always made, it's always around, but until it's stabilized, it can't act. ICE1 is a transcription factor. It binds to the promoter of CBF-DREB, that's our main cold guy. CBF-DREB is a transcription factor that binds to over 400 genes to turn them on or off in response to cold. The thing that all these genes have in common is a CRT-DRE element in their promoter. So they have a specific sequence in the promoter that says CBF binds here, and that gene is then transcribed. So you get transcription and translation, and you end up with a bunch of new proteins being turned on or off, and you end up with cold acclimation like that Arabidopsis that stayed green. So this is our pathway. This is the pathway my lab has been studying for a long, long time. My PI, he's been working with cold stress for probably longer than some of you have been alive in this room. So he's very, very familiar with this process. And we have turned our attention from Arabidopsis to soybean. Soybean is a super important crop species. It's actually the second most valuable crop in the United States. So you guys probably drive past fields of soybean all the time and never stop to appreciate how important that is to our economy. And not just to our economy, soybean can be used to make a ton of stuff. Yes, we eat soybean and tofu, we feed soybean to our animals, but it also can be used to make paint. It's used in biofuel. It's used to make clothing, sunscreens, crayons. The elevator of the Statue of Liberty runs on soybean hydraulic fluid. Right? Like, it's everywhere. So if we're going to lose soybean, think about how cold some of our winters have been and how cold some of our springs and falls have been. Soybean doesn't do cold. If we're going to lose soybean, that's a problem. It's very ubiquitous in our economy. When you put soybean in the cold, this is a time lapse that we took. So we're just going to watch our soybean. It's at control, now cold. And you'll see they start wiggling around a little bit. They're like, what's going on? The leaves start drooping. They don't like this at all. And then they start to actually like fall over. They start drooping after 2 hours. You get them in the cold for 2 hours, their leaves go down. Their leaves never recover. Right? As long as they're in the cold, they stay in this droopy, wilted-like position. If you pull them out fast enough, they'll recover. But they won't survive well. All right? So they really don't like this. And my lab is trying to figure out why. That's my project. Why is soybean cold intolerant? Can we figure this out? And can we do anything to help them out? Is there any way we could find some method so that we can help them survive cold stretches? We started this by looking at the bottom of this pathway. So genes that have those CRT, DRE promoter elements, right? We started at the bottom saying, well, are these genes that help cold, do they turn on? This particular family is called dihydrin. In Arabidopsis and strawberry, it's very, very important. Dihydrins are one of the major cold responsive proteins that turn on, and we're not exactly sure what they do, but they're very important. They go up really, really high, really, really quickly. In soybean, when we look at the GM ERD14 homologs, there's no increase in transcripts. They don't turn on. So something in this pathway is not functioning. Because at the bottom of the pathway, those genes and proteins that we need to make it cold don't turn on. So we took a step up. All right, the bottom of the pathway is not working. Let's look at that major transcription factor, CBF-DREB. What's going on with CBF-DREB? Do they even have CBF-DREB, right? Because nobody had looked at the genome. So my colleague, Dr. Yamasaki, that was one of his big projects, was he looked for soybean CBF-DREB, and he found a lot of DREB homologs, right? There are a whole bunch of these suckers. When he did similarities and looked at how closely do they align with the Arabidopsis versions, there were 7 that came with the Arabidopsis CBF. So these are the cold responsive CBF-DREB. And there were 7 soybean that seemed to be similar to these. So these became our interest genes, okay? What's going on with these 7? And it's hard to do 7 genes, so we only picked a couple to look at because we only have so many hours in our day. I'm sure everybody's familiar with that concept. And the first thing he did was say, do these turn on in the cold? All right, we know they're there. I can see them in the genome. But are they cold responsive? Because if they're not turning on in the cold, we need to go even farther up that pathway. Right? We're trying to figure out where the breakdown is in this pathway. They're there. If they're not on, we got to go a step up. So the first thing he did was look at transcript levels for a couple of these. Took the soybean, put them in the cold, and would take leaf samples and then grind them up and look at the mRNA copy number for these particular genes. And what you get is, this is 1 hour. In 1 hour, there is a gigantic increase in the transcript of these genes. This is one of the DREB homologs, and that's a 1,000-fold increase This is not a small system. This is a 1,000-fold increase of these transcription factors. And I don't have it next to it, but this is exactly what Arabidopsis looks like. Within that first 4 hours, you get a big increase in the CBF transcripts, and then they come down to near but not quite basal levels after the first 24 hours of cold. So we're seeing the same transient uptick in soybean. They're going up, okay? Well, maybe they don't work. Maybe they're not capable of binding to those promoter elements, and that's why they're not turning on their downstream genes. Maybe something has happened and they've mutated in a way that their DNA binding zone can't bind. Okay, well, how would we test that? So to test that, we actually went back to our old friend, Arabidopsis, because you can transgenically modify Arabidopsis. You can take any gene you want and put it in Arabidopsis. That's what makes Arabidopsis fantastic, right? Like, I can manipulate it every day if I want. This is going to be transcript fold change, and we're going to look at these 3 known cold CBF regulated genes. All right, so in Arabidopsis, they have that special promoter element. They are turned on. This is your wild type Arabidopsis before the cold, and this is wild type Arabidopsis after 24 hours in the cold. So all 3 of these genes are cold responsive. Their transcripts go up in the cold. The next thing that we did was we took those CBF DREBs from soybean and put them into the Arabidopsis without cold. So we had those genes with a special promoter that turns them on 24 hours a day, 7 days a week. They're always on. And we looked at these same genes in normal temperature Arabidopsis. So these should not be changing unless our soybean gene can turn them on, because the Arabidopsis cold system is off. And when we looked at them, that's exactly what we saw. For the 3 soybean CBF DREBs that we tested, all 3 of them turned on these genes in Arabidopsis. So that means these transcription factors are cold activated, and they're able to turn on downstream targets in a working cold system. Okay, now what? Now you have to start thinking outside the box. Now you have to start thinking outside your pathway. And that's when we came back to soybean. We're like, okay, transcriptionally responsive CBF DREB. Functional in Arabidopsis, it's still cold intolerant. Look at the amount of tissue damage here. What else might be going on? Are there inhibitory pathways in Arabidopsis? There's lots of literature about Arabidopsis. This is where you go lit diving. All right, you sit down with Google Scholar and you have yourself a little date and you dig around and you look for anything you can to try and get inhibitory. That brought me to IN3. All right, IN3 is known to inhibit the cold tolerance in Arabidopsis, and it's a little hard to see on this projector because it's a little darker, but if you see the green, when you knock out these 2 genes and you put them in the freezing without putting them in cold first, remember that very first picture of Arabidopsis we looked at where they all died because they weren't cold acclimated? If you knock out this IN3 gene, you get more green than you do in Colombia. So they have better freezing tolerance by getting rid of this IN3. Same thing when you look at the relative transcript expression, CBFs go up, CBF downstream targets go up without cold, just by getting rid of this IN3 and its homolog EIL. Just by knocking those out, you get an increase in cold response without actually having the cold. So I was like, ooh, okay, let's see what this guy's doing. So IN3 turns out to be a major transcription factor of the ethylene signaling pathway. If you think you're not familiar with ethylene, I guarantee you are. If you've ever had a banana that was perfect and you went to bed and your banana was deader than dead, you have experienced ethylene. Ethylene is a growth and fruit ripening hormone. So it turns on all of the ripening, turns your tomatoes from green to red, your bananas from not ripe to perfectly ripe to, ew, I can't touch that, or that has to go into a cake. You have seen ethylene. It's a very small molecule. You don't think it would do much, but it's involved in everything from seedling to fruit development and ripening to even some stress responses like we just saw with Arabidopsis. We're going to take a very brief look at the ethylene pathway because that's where I'm now headed. I've left CBF and I've moved into the ethylene pathway to see if it's interfering with CBF. With the ethylene pathway, you have a membrane receptor that's ETR1. CTR is phosphorylating ion 2 when there's no ethylene. So without ethylene, this phosphorylation is keeping this system turned off. When ethylene binds, the receptors actually change shape. This C-terminus is cleaved and it moves to the nucleus. So it actually leaves the ER membrane and translocates to the nucleus, where it stabilizes ion 3. And nobody knows if this stabilization is direct, like ion 2 is directly stabilizing ion 3, or if ion 2 is turning off EBF 1 and 2, which cause ion 3 to be broken down. It could be either way, nobody knows, but the end result is ion 2 stabilizes ion 3. Ion 3 is our transcription factor we care about. In the ethylene pathway, it's meant to go turn on ethylene. In the CBF pathway, it negatively regulates CBF. And that's what I care about, right? The fact that ion 3 is known to negatively regulate. And since I know something is happening here in soybean, this seemed like a good place to check. We are fortunate enough that my colleague, Dr. Yamasaki, left me some wonderful RNA-seq data. And if you've never seen RNA-seq, it's basically I have 50,000 mRNA levels matched to their genes, so I know which gene they are, like which transcript is being made, how many copies of it, for the entire first 0, 1, and 24 hour of cold exposure in soybean. So I've got a lot of data I can mine. And that's exactly what I did. I went into our RNA-seq data to look at some of these key genes in this ethylene signaling pathway to figure out, are they being regulated differentially than they are in Arabidopsis? Because this is a negative regulator. If it's going up, it would make sense that soybeans cold is going down. And when I look at it, that's exactly what I see. Ion 3 is the darker the red, the greater the upregulation. And ion 3 goes pretty red. Ion 2, its positive regulator, also is going pretty red. And EBF1, 2, its negative regulator in early cold is going down. So it's not being negatively regulated, it's being positively regulated. In Arabidopsis, this doesn't happen. These genes do not change in the cold. So it might be a big deal that in soybean, these genes are changing. They're going up, so that this negative regulator may be going up. How do I test that? Well, thankfully, there's this thing called literature. And we know that, from the literature, we know that silver ions block this receptor. If you turn off the receptor, doesn't matter how much ethylene there is, nothing's going to happen downstream. Right? So if we turn off the receptor, we're basically turning off ion 3, our negative regulator. So we're going to spray silver on our soybean. The problem with this is there was a lot of research for seedling development and ethylene response, seed development and ethylene response, but not a lot for baby soybeans, which is where I work. I'm interested in the seedling stage. So before I can even do cold response, I have to make sure that they will respond to silver in the way I anticipate. It's very important that you do your pilot studies and you know things are going to work the way you're expecting them to work before you do the big money experiment. Because what if it didn't work because silver doesn't work in soybean? You wouldn't know that. So the first thing that I did was I looked at this ethylene signaling pathway and I took these little soybeans, cut them and put them into test tubes that had either a 1.38 millimolar ethifon. This is just quickly changed into ethylene by the plants. So you can just think of it as I'm feeding the plants ethylene, just giving them a whole bunch of ethylene. Then I'm going to spray some of them with silver nitrate and spray some of them with water. And I'm going to look for signs of the ethylene response. And in this case, we're going to look at cotyledon, which are these little seed leaves. Ethylene exposure makes them abscise or die and fall off. So it's a very easy assay. I go and I look. Are they still there? And then I come back the next day. Are they still there? And I'm just literally going to count, did they die and fall off? When we do this for a week, these are all in water, and these are in our synthetic Ethifon. All right, so you're going to look at the cotyledons. This is just day 1. Everybody's green. Everybody's happy. That's what we'd expect. Day 3, you're going to start seeing some yellowing. These are the ones being fed our ethylene. So they're starting to yellow. By day 7, they are gone. Completely gone. If you look at our ones fed Ethifon and fed silver nitrate, there are some gone and some still there. I've done this 4 times now, and so we can look at it in graph form. You just have percent cotyledon abscission, how many days they're in that ethylene. Without silver, you get pretty good abscission. With silver, you get less abscission. Awesome. So I can delay ethylene from impacting these plants. But can I completely get rid of it? That became question number 2. Okay, I've delayed it. What if I spray silver the day before I put them in that ethylene? So I preemptively block all the ethylene receptors, and then I feed them external ethylene. Can I get a better result? And that's exactly what I did. Now, unfortunately, I didn't think this through, and I took these pictures in a different orientation the second time I did the experiment. Don't do that. Think it through and bring your lab notebook with you when you're setting up something so that you don't have to go and be like, yeah, so now ethylene is over here, and now, yeah, just think through these things. These guys fed ethylene, sprayed with water, fed ethylene, sprayed with silver nitrate, and this is day -1. That means they're in water right now, right? So the day I started the experiment, everybody's been sprayed, but everybody's in water. The next day, I moved them to ethylene, and 3 days in, they're all gone again. All of the cotyledons are still here. Day 5, all gone, all still here. Look at the graph. Hello. Only the ones I fed ethylene and sprayed with water lost cotyledons. The ones that were fed ethylene and sprayed with silver the day before never lost their leaves. So I can completely block by spraying 1 day before ethylene exposure. If our hypothesis is that cold is creating more ethylene signaling, spraying 24 hours before we put them in the cold should block or dampen this process, right? That's our hypothesis here. And when I looked at this, this is telling me ethylene signaling. I dialed in to that IN3 transcription factor. So this is qPCR, transcripts just of IN3 in soybean, number of copies. And on my 4-day, here is fed ethylene sprayed with water. Here is fed ethylene sprayed with silver. I can block the increase in IN3 caused by ethylene exposure with this method. At this point, I felt pretty comfortable that we could actually do this in the cold and see if what we saw would be based on ethylene signaling being dampened, because I just proved that I could do that in soybean. Our question then became, is the ethylene signaling pathway, likely through IN3, regulating cold tolerance via negative regulation of CBF-DREB? If I'm correct, blocking with silver should lead in a decrease in IN3 and an increase in CBF-DREB and cold tolerance. So one of the tools I have at my disposal to do this, because qPCR is expensive, time-consuming, and really annoying, is a transgenic soybean line that expresses an abiotic stress reporter. So in this line, we have the Arabidopsis RD29A promoter driving GFP-GUS. We call this abiotic stress responsive because it has 2 responsive elements to wound, 1 to drought, 1 to the phytohormone ABA, but 3 to our CBF transcription factor, the one we're interested in. It has 3 signals that CBF can bind at any of these 3 points. There's also a herbicide resistance gene on the plasmid that gets transformed into soybean. This is important because it means only the plants that have my reporter gene in it are resistant to glufosinate. Any plant that didn't take up this gene will have damage due to my spraying this herbicide. So this lets me pick and choose only the plants that have my gene in them. And I'm going to show you the arduous process that this is. This is a leaf. I paint the herbicide, whoop. I come back in 5 days and I look at them. It is super hard. Minimal to no tissue damage. They're considered resistant, severe to moderate to severe tissue damage, susceptible. And I just count them. I'm looking for Mendelian genetics, a 3-to-1 ratio to let me know that 1 will be homozygote, 2 will be heterozygote, have 1 copy of my transgene, and 1 would be wild type, it would be susceptible. And I found 3 out of my 5 lines that sorted that way. So I moved forward with those, and I wanted to find that 1 plant that would have been homozygous, right? I want it to have 2 copies so that I know it's going to behave the way I expect it to behave, and that any reaction's not due to genetic differences between my plants. I want them all to have the exact same copy number. I did find 3. I'll tell you that picture and this picture were a year and a half of my life, because soybean takes 4 to 6 months for every generation. So I found 3 that were 100% resistant, but it took me a year and a half of my PhD. Soybean is awesome, but annoying. Now I'm finally ready to say, if I spray them with silver, what will happen? We're a year and a half in, by the way. I'm finally getting here. I'm going to measure this with the GUS part of my transgene. So GUS is an enzyme, beta-glucuronidase, and it breaks down complicated carbohydrates into simple. In this case, the simple MU glows. It's a fluorescent. So as GUS is activating, it's taking non-fluorescents, and fluorescents will be generated. Because every time it cleaves a MUG molecule, it's releasing the fluorescent MU molecule. So I can measure relative fluorescence over time, and the slope gives me the activity level of my enzyme. So that's what we're going to look at in the next graph, the slope, the activity level in GUS. I'm loading the exact same amount of MUG and the same amount of total protein. So the only difference between my samples should be the ratio of GUS to the rest of the total proteins, right? If there's more GUS being made by the plant, it would be more present, so we'd get a higher slope. Less, we're going to get a lower slope. And that's what we did. Took these guys, sprayed them with silver nitrate the day before and the day I put them into cold, left them in cold for 2 days, looked at the GUS activity and my different conditions. Here's water sprayed, 4 degrees. Here's water sprayed with silver. It's a 2.5-fold increase in activity level for all 3 lines. So just by spraying silver, it went up 2.5-fold. This is my end target, right? GUS activity is activated by CBF. So I assume that means CBF is being more activated, but we don't assume in science, we check. So I went and looked at CBF transcript levels. Using qPCR, I looked at relative transcription levels, and again, water, 4, and silver, 2.5-fold increase of CBF transcript level. 2.5-fold increase of GUS activity. I like where this is going. But that's just molecular. Doesn't tell me anything about how they're going to do in the field. So we have another assay. It's called electrolyte leakage. And what it does is it lets us measure the cold tolerance. You take your leaves, you cut little circles, you put them into the test tubes, and every 2 hours you drop them down by 0.5 degree until you get to whatever temperature you want. Right? So it's a slow freezing process. As damage occurs, plasma membranes are going to break, and that's going to release ions from inside the plant cell to the outside. That's what I measure. I use a conductivity meter to measure the amount of ions that are in the solution. The more ions in the solution, the more damaged they were by the cold. And I can look at this over a time course, which is what I did, of -1 to -4. And you just got electrolyte leakage percentage. So unlike a lot of our values, in this one, low is good. We're playing golf. All right? Low numbers are good numbers. You don't want high scores. High scores like are 22 degrees before, so non-cold acclimated, right? Held at 22 degrees, sprayed with water. When we have Spray with silver, we get a marked decrease in electrolyte leakage or an increase in freezing tolerance. So by spraying with silver, we increase their freezing tolerance. And we get some of that on our cold acclimated plants. But after 2 days in the cold, they've acclimated as much as they can. So this is good for hitting them at the beginning. Might not be so good for a long-term cold exposure. But when you look at these guys, they sure look a lot better, right? So 4 degrees, before I cut the leaves off to do the experiment, sprayed with silver, and then the one sprayed with water, they certainly passed the eyeball test. This phenomenon only happens at low light. I actually observed it because one of our bulbs went out in the growth chamber, and I had 100 micromolar instead of 200 micromolar. It doesn't happen at high light. We're trying to figure out why, but it's still interesting to note. Science is never done. So silver blocks perception, right? It blocks that receptor. Well, let's really make sure this is ethylene, because silver can do other stuff in the plant too. We're going to use what's called AVG. This is the synthesis pathway for ethylene. It starts with methionine, goes through a couple steps, and you get ethylene. AVG blocks ACC synthase, which is the rate-limiting step. So this is that important step. We're just turning that enzyme off, and we're going to see what happens to our plants doing the same experiment. Spray them with AVG, put them in the cold, measure the GUS activity, 2.5-fold increase. Spray with water, spray with AVG when they're in the cold. Just 2.5-fold increase of our CBF, 2.5-fold increase with silver, 2.5 with AVG. So I'm still pretty confident that this is what's going on, right? Ethylene is negatively impacting in soybean. However, to really get this hammered in, I've got plants right now growing, so I can repeat that electrolyte leakage assay that I showed you with silver with AVG, 1-MCP, which is another inhibitor, Ethifon, that stimulator that turns into ethylene, and then ACC, the precursor to ethylene, to see what happens. So I've got both turn ethylene on and turn ethylene off. And look what happens to both the freezing tolerance and that GUS activity. I want to look at all the transcripts that I can for the CBF, the IN3 family, and then their downstream targets, so I can really hammer home what's going on in this pathway. Just want to make sure all those connections I think are happening is really, I can demonstrate that they are happening. So to sum up what I hope you take away from this, the ethylene signaling pathway inhibits cold response in soybean. I know this because when I inhibit perception of ethylene with silver, I get an increase in my reporter activity, I get an increase in CBF transcript levels, I get an increase in freezing tolerance. And when I block ethylene biosynthesis, I get an increase in cold reporter. So we can increase soybeans' cold tolerance by treating them with something that turns off ethylene right before a cold. If you're a farmer that's planted a whole bunch and you hear on the news, we're getting a weird late frost, there go all your soybeans. But if you could put some of this stuff into your irrigation system, give them a good spray, and they'd survive that weekend, that would be huge. So that's what I'm trying to do. Science does not happen in a vacuum, so I need to say thank you to a couple people. First and foremost, the most adorable lab helper on the planet, my son Wayne. He comes in with me midnight. He has been in my lab because I've had to take samples. He helps pipette. This is him helping me with one of those electrolyte leakage, putting the DI water in all the tubes for mommy while mommy's doing something else. And he racks pipette tips, right? So he's all over our lab. Past and present members of Randall Lab, this is my boss right here, Steve Randall. He is awesome, the most supportive boss I've ever had in my entire life. If you're looking for a PhD or a master's, he's awesome. Seriously, he's super awesome. United Soybean Board and the IUPUI Department of Biology, because they pay me. I like being paid. Of course, Dr. Wyman for inviting me, and I'd like to thank all of you for your kind attention.
Questions before Dr. Baravich is excited.
I know it's becoming more popular for people to be more skeptical of treating plants with different things or GMOs. Do you think it'll be difficult to begin different treatments of plants are coming up with more genetically modified plants in order to combat these climate changes when people are so against those kind of changes?
I think we have a harder sell with GMOs than we do spraying because we're used to spraying our plants with things, right? People go and they spray pesticides, herbicides, all sorts of chemicals. So there's a chemical that I just finally got my hands on called 1-MCP. It's already commercially used to treat vegetables to make them ripen faster. So, it's already used in the agriculture industry. So if it works the same way silver does, I think that will be a really easy sell to people because they're already eating it, right? It's nothing new.
You just spray the plant once with silver, you don't spray it repeatedly?
I spray them day before and the day they go in the cold, and then I do not spray them again.
What do you know about transport of silver through the vascular elements of the plant? Is it transported effectively?
It is transported effectively. That's one of the things we're worried about with off-target effects because I'm spraying it on the leaves, but we don't know what it's doing in the roots.
Here's my next question. You're growing these in kaolite, or what are you growing these in?
I have a growth chamber, and
No, the
it's
soil. What soil?
oh, ProMix. It's potting soil.
Okay, are you getting interactions with binding? I'm just wondering if, because depending on the soil type, you might have cation exchanges with the soil and stuff like that.
Yeah, we haven't looked at this particular soil for that. This is our standard potting soil, which is why we use it, but we kind of have that in the back of our heads. I've done not the cold experiments, but the other experiments with a couple other cations and ions to try and see if that they are having an effect, and I don't see one, but I can't say for certain yet.
Do you know what ethylene does in soybeans?
So the big thing ethylene does in soybean is it causes the seeds to ripen. So pod development, that's why there's a lot of research from flowering to pod development. When ethylene signaling is turned on and off is very critical so that you get high yield. So that's the big thing it does. It also, when the cotyledons come up, ethylene's responsible for that motion that pulls them out of the soil and moves them upwards. So it's really important at the beginning and at the end stages of life.
So would the silver blocking the ethylene make any difference for like soybean production? Or is it not like long enough?
It's not, it's a short-term exposure, and I would never, ever, ever, ever say spray your field with silver because like that has so many other implications. Silver was my sledgehammer. It's cheap, it's easy, and I had a ton of it lying around the lab. And that's why I did it again with AVG and why I was working to get that MCP, which is already used for agriculture. But if the sledgehammer didn't work, I didn't want to spend a whole lot of time on the more expensive, hard to get chemicals.
How many undergrads have you worked with on this project?
This project, I have 2 that have been on this project. And one, we did more cold acclimation, not silver, not the silver side. And that paper just got accepted for publication. So it'll be out later this year. And she's on it. She did quite a bit of the electrolyte leakage work. So.
Other questions? Well, thank you so much for coming today. I truly appreciate it. Again, you're welcome to have lunch later today. For one more hand, if you please.